Good morning, everyone. Today I'm just recording, or this morning I'm recording this short lecture on bacteriophage for you to watch sort of in preparation for the lab this week. So in the lab this week, since the topic of this week in microbiology class is viruses, I wanted to talk about bacteriophage a little bit because we're not going to talk about it a lot in the lecture. And the topic of the lab is on phage therapy. So I wanted to make sure you had a little bit of understanding. And particularly, we're going to talk about Bacteriophage T4, which is the topic of one of the papers that is in the lab. So let's switch over to sharing the slides. And this is Bacteriophage T4. Again, a bacteriophage is just a virus that infects a bacterial cell. So this is an example of a virus that infects a bacterial cell. And you immediately see that the structure of this virus is actually much more elaborate than most of the viruses that we've looked at in class this morning. You'll see some of the diverse morphology of viruses, and there's some diversity in phage morphology too, but this is one of the most elaborate. So it's a multi-part virus that has sort of the capsid up here with the genome in it, and this is, you know, a sort of a classic icosahedral capsid structure here. So this is made by proteins. There's no envelope on this. But then we have this elaborate structure down here. So there's sort of a rod here, and we call this the tail structure. And the genome is going to come down through this and into the bacterial cell. And these tail fibers here, which look amazingly like a spider or like a lunar lander or something, are going to be the site at which the phage contacts the bacterial cell initially. So these are sort of the receptors. And so we'll see in the next slide how this is going to work. So the genome of this phage is made of double-stranded DNA. It's a pretty large genome for a virus. It's almost 170 kb, kilobases of DNA, and about 160 genes in that genome. So pretty large and complex, even though it's infecting a bacterial cell. The receptors that this virus is going to recognize through the tail fibers and down here on this base plate are going to be the lipopolysaccharide in the outer membrane of the E. coli target bacteria, and then the OMP-C porin protein. So you remember the porins are outer membrane transport systems in the outer membrane of gram-negative bacteria for facilitated diffusion. So obviously they're exposed on the surface of the cell, and so this is a molecule that the bacteria is going to recognize as well. Okay, so it's going to bind to the surface of the cell and inject its genome into the cytoplasm. So this is what that looks like. So the tail fibers attach. So you see the receptors that it's contacting here are the porins in the outer membrane. There's a conformational change in the tail fibers that bring that base plate down into contact with the surface. The tail proteins allow the inner core protein. So inside this thing that looks like sort of a spring structure here, these proteins are going to extend through the outer membrane and the peptidoglycan, ultimately contacting the inner membrane of the cell. So the plasma membrane, when that inner membrane is contacted, that initiates the transfer of DNA through this channel and into the cytoplasm of the cell. So it's pretty amazing that this structure is ultimately going to be able to extend through all the way through the cell envelope to release the DNA in the middle. I'm not an expert on all the studies that have been done on T4 over the years, but this seems like a remarkable process of protein rearrangement. There's no ATP to drive any of this, right? So this is being triggered here by contact with the cell surface to undergo a fairly dramatic rearrangement to generate a channel for the DNA into the cytoplasm of the cell. Once that DNA gets into the cytoplasm, this is sort of what it looks like. So again, the main body of the protein never comes in, the main body of the virion structure never comes into the cytoplasm. It's just the DNA. This is actually the basis for a fairly famous experiment showing that it was actually the DNA that's the genetic material. And this is called the Hershey-Chase experiment. We'll talk about this later in class. So it's the DNA that's genetic material and not the proteins. Okay, so the DNA is injected in. It starts, it has genes on it. The genes use the host machinery as well as their own transcription factors to stimulate their expression using the bacterial RNA polymerase. So the viral proteins start being made. One of the things that happens then is that the virus directs the host DNA to be chopped up because it doesn't want the host genes competing for the RNA polymerase and ribosomes, etc. Right? So the phage DNA is then starts to be replicated. More genes are expressed. There's a whole sort of genetic program to make the genes so the genes are expressed in the right order to make the proteins in the right order, right? And then you see here the viral heads and tails start to be made. The heads are filled with newly replicated viral genomes, and the virus is assembled inside the cell, and then the host cell is lysed. Now, as you might imagine, lysis requires breaking the host peptidoglycan. So one of the products of the bacterial genes is lysozyme, right? So phage lysozyme to eat away at that bacterial cell wall so that it can be broken to release the new viral particles. You can see down here an electron micrograph showing sort of to scale, since this is actually a picture, the formation of new phage virions, the heads inside of the host cell. In fact, they sort of aggregate into regular, almost crystalline structures inside this host cell. There's so many of them being made. So hundreds at least of new phage particles being made that are then going to be released when this virus breaks open, when the host cell breaks open. Okay, all right. So quantitation, this is another thing that's talked about in the lab today. How do we quantify the amount of virus that's there, viral growth, etc. For many viruses, that's not easy. For some phage, at least, it's somewhat easier because they kill the host cell. So you can do what's called a Plaque assay. So the way that viral particles are typically measured is as a titer, a concentration of infectious viral particles or entities or virions per milliliter, say. It turns out that a lot of viruses, when they are made, they're not necessarily active. So this isn't necessarily the same as the total concentration of viral particles. It's the number of infectious virus particles, right? So the methods can include ways of counting the total number of viral particles, if you have a way of doing that. For example, if you have fluorescent antibodies, so you can light them up in a microscope, that kind of thing. Or you can do a functional assay, like we were just saying, of a Plaque assay. In order to do a Plaque assay, you need the susceptible bacteria, or if this wasn't a bacteriophage, if it was another kind of virus, you might need cultured host cells that could be attacked and killed by the virus. Okay, so a Plaque assay, and again, you're going to have videos to watch on this. The way you do it with a bacteriophage looks like this. So you have a starting culture of live bacteria. They're going to be your host. And then this is actually some melted agar. It's a low concentration of agar. So it's actually going to be used as an agar overlay for this. So what we are going to do is we're going to have our phage sample here. This shows a large amount, it could be a small amount, whatever, doesn't matter. You just have to know how much of it is there. And then you're adding the bacterial host cells that the phage is going to bind to and attack, right? And here it doesn't really matter how many of them you add. You actually want to have a lot so that every viral particle can easily bind to a host cell. You want to have a lot more bacteria starting out than you have virus, okay? And then you add this melted agar. Again, it can't be so hot that it kills the bacteria, right? So we usually keep it at about 40 degrees Celsius so that it'll stay liquid, but when we add it to the bacteria, it won't kill them. And what we do is we then pour that whole mixture of virus, bacteria, and melted soft agar onto the top of a regular agar plate. Okay, so this is actually forming sort of an overlay on top of that. And then we let them grow up. We stick them in the incubator and let them grow overnight. Okay, so typically what we do with this phage sample is we actually do a series of dilutions because we don't know how much live virus there is to start with, right? And so what we're ideally looking for is plates where it's mostly covered with bacterial growth, but everywhere we started out with one virus, the virus has started to grow by killing the cells and then killing the adjacent cells when it's released. And the point of the soft agar here is to keep the virus from just spreading everywhere really fast, if it was liquid, right? So it just spreads a little ways to the next cell, kills that one, and you get sort of a spreading area of dead cells. And that's a plaque, right? So each one of these plaques is inferred to have come from one starting virus. In the same way that when you're streaking a plate, each colony is assumed to have come from one live cell, right? So on this sample here, and we can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. There's 14 infectious virus particles at the start of this plate when we mix them with bacteria and spread them out, right? So if we know what dilution that was, then we can extrapolate from that to what the concentration of viable infectious phage was at the start. Okay, so that's the Plaque assay. Now that only works if the virus actually kills the bacteria, right? So that we can see this cleared zone where there are no bacteria, right? The problem with that is that not all phage do that really cleanly and efficiently. There's two ways we can think about bacteriophages. One is as a lytic phage. So every time they go through a life cycle like T4, they're going to replicate and make new viruses, burst open and kill the host cell, right? But there are also what we call lysogenic phage. And lysogenic phage actually can integrate into the genome of the host cell and sort of go dormant so that they're basically replicating with their host cell. To a certain extent, this is what a retrovirus like HIV does as part of its life cycle. It integrates into the chromosome. Some prophages don't even integrate into the genome. They actually replicate as plasmids. Often what they do is they have sensory systems so that if the host cell gets in trouble, like if the host cell experiences stressful conditions, then the phage are triggered to say, this guy might die. I'm going to get out of here. So then they transition back into a lytic life cycle, right? So lysogenic is not necessarily a permanent state. It can be, a transient state. But we find phage genomes, and we call them prophage when they're integrated like this, in pretty much every bacteria that we look at. So we know that it's a common strategy for viruses that infect bacteria. Now, T4 doesn't do that. T4 is only lytic. It doesn't integrate into the host genome, right? So that's an option as well. But that means it makes nice plaques so that we can see it really easily. Okay, I'm going to stop sharing here. That was our introduction to bacteriophage, particularly the T4 phage, and I will see you in lab this afternoon.